tell me what you guys said about what you know about DNA.
Well, first, it's like it like encodes like genetics, basically.
What's that mean?
It like traces back to your genes.
So what's bigger or smaller, a gene or DNA?
What do you mean? Genes,
How many of you agree with that statement? Genes are smaller than your DNA.
You said that, not me.
Well, doesn't DNA hold genes? Isn't that a thing?
How many of you know? How many of you are confident you know? How many of you are willing to pick a fight with them about that statement?
You can pick a hundred, not me.
There's a reason that I wrote it in quotes, and it's not anything to do with the people I've called on, and everything to do with the perception of the general public. This is where the vast majority of the general public misunderstands what DNA is and what genes are. So that was a good place to start, so we can make sure before you leave this room, we are perfectly clear, because I certainly can't have you go out there with the rest of the world and understand things like the rest of the world understands them. You're by far better than that. What else do you have written? Our team in the corner would like to not have the heat anymore.
No, we like heat.
One moment. They like heat. Okay?
Corner. It's a double helix.
Nice.
What's that mean?
It's like a ladder shape twisted.
It's a model made by Watson and Crick in 1953.
Exactly.
Oh,
what I
yeah.
said.
That's why I remember it
How do you find that out?
from the notes we were supposed to.
Nucleotide, nitrogen base, monomer, polymer, biomolecule. We're paying those a much closer month-long visit now. Yes, if you think you just heard me say month-long, you are correct. DNA is from now until final exam in 4 chapters.
Is there a final exam in stuff in like first semester too, or you just?
Nope. There's too much to remember.
Yeah, that's what we did at St. Mike's, and it was awful.
Yeah, that's...
Especially
The synonym I would use for cumulative over the course of the year is bloodbath. Seems accurate, yeah? I mean, I've never tried it. We could try it if you want, so I can test my accuracy.
pretty accurate.
You guys are like, no. Some hypotheses do not require testing. Are you sure? You've all probably been told it's the stuff you get from mom and dad. That's a popular one here. Stuff you get from your parents. Stored in what organelle? The nucleus. Instead of from mom and dad, sometimes students tend to say unit of heredity. It's what you inherit. Any of this stuff sound terribly new yet? A lot of this stuff, I think that you guys have a pretty good foundation in it, with just a few minor misunderstandings that, again, not likely your fault, likely the result of some oversimplification of previous education and oversimplification of the concept to make a difficult concept more accessible. A little bit of history. I give you this little bit of history, and you notice that I say woman. A little bit of history. The idea of inheriting something from parent to child in biology is not necessarily new, but it became something acceptable around late 1800s into the early 1900s. But the problem is no one knew how. So that was the big question, not a clue. It was 1950s when they finally were able to identify the fact that that particle that was passed on from generation to generation was in fact DNA. They gave it the name DNA. And researchers spent a great deal of time trying to elucidate the structure, to figure out what shape it is. And there are two scientists who are often credited with doing so in 1953. Their names are James Watson and Francis Crick. And you will not go to a biology class and not hear their names. What they found in 1953, or what they published, I should say, in 1953, was a huge deal.
Didn't she find it and then they published it before she did?
They stole it. Straight up stole it. So yes, indeed, that's why Watson and Crick don't get their picture in this particular slideshow, because quite frankly, Watson and Crick, she worked in their labs and they took the X-ray crystallography that she did of that structure. And one of them has since repented and then died. Apologized for having done so, and the other one, until his deathbed, would not give her credit. He was not really an upstanding kind of guy.
Which one?
Watson would not give her any credit. Rosalind Franklin. So Watson and Crick are given credit in 1953 for elucidating the structure when it was really this photograph, that plate right there. You're looking at it and you're like, that doesn't look anything like any DNA molecule I have ever seen, right? So that picture is what revealed to them, based on the grooves in the side of it and the size and measurements of all of those grooves, what exactly that looked like. And this is Rosalind Franklin. And sometimes it's a little bit hard for us now to understand why this is such a big deal, why biologists fuss about it, but remember, it was 1953. She was a woman and Jewish, therefore doing something a lot of people did not want her to do. Think about it. 1953 comes right after what? Yeah, so she was fighting a very hard battle. Luckily, she, at least gets credit in most textbooks.
Okay, so did Watson and Crick find the model of the formation of the DNA? So she founded the DNA?
She took this photograph, and based on measurements between the marks on the photograph and what they already knew about it, they were able to infer the shape of DNA based on what we call the minor and major grooves in the side of it. They compared the measurements. She took the photo. They interpreted the photo, but they took the photo from her. So it's kind of how it plays out in a fairly ugly way. It was not until, my lifetime, they started a project called the Human Genome Project. So 1953, we figured out what the structure of DNA was. 2000s, they set about trying to map where every single gene, every single nucleotide is in the human genome. Genome is just the fancy word for every gene an organism has. Actually, more specifically, every nucleotide an organism has. In 2001, they completed that. In 2001, they completed it. So that took like a good 50 years, right? It's a long time. What that means for you guys is 2001, scientifically speaking, is alternately recently and ages ago. It's fairly recent compared to the other stuff we studied. Like, think about good old Charles Darwin. That was like 1859, right? And then this, we're talking like 2001. So in comparison, this is new. But what I will tell you is what it means is a lot of what you are going to learn from now until June is still changing. A lot of what you're taught here is going to be very, very different even in the next 5 years. So those of you who are working on data and design this summer, this is where I'll tell you, you are the scientists who are going to be shaping what that looks like. That's our goal for you, is to put you in to real science where you will make a real difference in mapping genomes. There's a lot of readily available data. You just have to write the software. So that's what we're looking at. That's how new this stuff is. And again, that's part of why there's probably a lot of misunderstandings. You were taught by, how old would you say your previous science teachers were?
Pretty old. Well, she wasn't that old, but she looked older.
She was old.
You guys made her look older?
No, she made herself look older.
Harsh.
How many of you would say, now keep in mind, I've been out of school for 8 years. And the only reason I know the recent data on any of this stuff is because I make it a point to go to those things at the NAS with Mr. DF, right? Like, this is what we do when we're there, is keep current on this. How many of you are like, yes, my previous science teachers graduated from university within the last 8 years and keep current on all of this research?
My old science teacher went to the Maroon 5 concert a couple weeks ago.
Oh, good. That's super helpful. So there's one of you who are like, yes, my previous science teacher would qualify as that. Here's where I'm going to say, in our misunderstanding during our brain dump, this is not their fault. It's because this is how they, like, that misunderstanding is what they were taught. This is the very basics of everything that we're going to talk about from now until June. That DNA is at the center of this great big picture. So we do call it the central dogma of biology. Center, right? Middle. Dogma, foundational principle or belief. So at the very center of everything we believe in biology, DNA codes for RNA. So DNA codes for RNA. And RNA is going to code for protein. That basically means anytime you've ever been like, oh yes, my DNA is it's a code for genes. All really it is consider your DNA a recipe book. You've all seen a recipe book, yes? It's just a series of instructions for making separate recipes. Turns out here the product happens to be protein. You're just a giant set of instructions for proteins. All different kinds of proteins. How many of you object to that? You're like, I am more than a pile of protein. I have a bunch of reductivists in here. You're all like, no, really, if you just keep taking me, like breaking me down into my little parts, my smallest parts are proteins, the smallest functional things. If we're in a Catholic school and I have a room full of reductivists, we have a problem, just so you know. Big problem. Don't go down that road. Don't assume that because I'm telling you that DNA is the most important biomolecule or information molecule, that it is the essence of who you are. It's not what's being said here, and that's a gross misunderstanding that can get you some pretty dangerous places. You know this one. This is a gimme. Newt, it's such a gimme, you're going to fill out the rest of this chart for me. Tell me what to write down.
Monomer, simple, polymer, not simple.
But specifically for this kind of molecule. What's the monomer of a nucleic acid? And the polymer, there are two choices, two kinds.
Nitrogen base.
This was October. So this is where we traipse back through information. Your monomer here is a nucleotide. Your polymer is either DNA or RNA. The one we're going to focus on first, this is the chapter, just DNA. You're accustomed to the cartoon version of this. We're going to look at the actual chemical version of this, not the cartoons. There's nothing interesting about the cartoons. Do you recognize that thing? Where this is a phosphate. This is a 5-carbon sugar. And that is a nitrogenous or nitrogen base. Yep.
So that's the monomer of DNA.
This is the monomer of DNA. So if we're talking specifically about a human, your DNA is made of 6 billion of these. This is one of 6 billion. There are 4 choices that we can attach at where it says base, the nitrogen base. We can either attach an A, a T, a C, or a G. So we can attach either A, T, C, or G. The sugar is always going to be the same sugar if we're talking about DNA. If we are talking specifically about DNA, the sugar is deoxyribose. And the phosphate's always phosphate. Six billion makes up one human. And I don't mean 6 billion scattered throughout you. I mean 6 billion in every single cell you have. There are 6 billion nucleotides. Actually, not even in every single cell. In the nucleus of every single cell you have, I like to narrow it down, be very specific. In the nucleus of every cell you have, there are 6 billion nucleotides. So these must be pretty small, right? So if we're looking at the 4 different possibilities of things to attach, here they are. That's the one I labeled C, cytosine. This is thymine, adenine, and guanine. For the purposes of discussion here on out, A, T, C, G, right? Just the first letter, easier on everybody. But this is what their structure looks like. Yep.
Do we need to know the structure?
You will need to know how many rings their structure has. You will never be asked by me to make or replicate that ring. I will never be like, yes, draw with all of the double bonds in the correct place a thymine. I won't even ask you to label them. I do want you to know which ones have 2 rings and which ones have 1 ring. It's not necessarily a super simple way to remember it. There's one thing that I remember it with. When I say ring, I mean, sound effect coming now. This is 1 ring, right? This is 1 ring wide. Remember when we talked about biomolecules, we said carbon chains have the unique ability to fold back on themselves into a ring shape? That's what happened here. We had a chain of carbons that folded into a ring. For adenine and guanine, there are 2 of those that folded back on themselves. I've always remembered it in this way. We classify our nitrogen bases quite simply by the size of their shape. So whether they are one ring or two ring. The one ring nitrogen bases are labeled as pyrimidines. Will I expect you to know that? Absolutely.
So the one rings are pyramid.
The one ring structures are pyrimidines. I always remembered it that the longer word got the smaller structure. Like if I had labeled them, I would have put the shorter word with the shorter structure because I like that sense of organization. But clearly they didn't give me an opinion. So the longer word goes with the longer structure, the shorter structure. And the shorter word, purine, goes with the bigger structure. So two-ringed nitrogen bases are purines. Yep. So these attach right here where it says base. So the whole nucleotide is going to be a combination of phosphate, sugar, and then this great big structure will attach there. How many of these did we say in one person? How do they package up? How do we make them fit? Here's your double helix. And I want to give you a little bit of perspective because this addresses our issue we started the hour with, the gene being smaller than DNA. Here's one nucleotide. See this red thing? That's supposed to be the nitrogen base. And this gray thing is a combination of sugar and phosphate. So here's one single nitrogen base with one that's complementary directly across from it. There are two spellings of complementary. What are they? These two nitrogen bases are complementary in that they add up. They fit well together. They fit like perfect puzzle pieces. That's an important part of DNA structure. So they will fit like a perfect puzzle piece. Now they always fit two bases directly across from each other. So if there are 6 billion total, how long is this whole thing? If they're always paired up directly across from each other. So it's 3 billion long. It's a chain of 3 billion. Here's two more. Here's our DNA. It's going to curl up around histones, right? As we said last chapter. And a histone is going to make a complex of 8 of them and make a nucleosome. Those curl up into a little pretty little flower shape. And that pretty little flower shape stacks neatly on top of it to make like a rope. That rope is going to squiggle up and keep squiggling until we get to this thing. That perhaps if I trace it, what is that laying on its side?
What's the flower looking thing called?
You can call it, well, it's nucleosomes, and then they make a chromatosome, but I don't know that I'm too worried about you knowing the name of it, just that it packages further. The reason I point this out to you because it addresses our issue. A gene is typically, well, let's give it an average. A gene in a eukaryote is typically about 1,000 to 2,000 nucleotides long. So what's bigger, a nucleotide or a gene? Gene is bigger, right? Because a gene is made out of 1,000 to 2,000 nucleotides. What I mean to tell you then is that that would be one gene. Yep.
So, like, a bunch of genes make up one DNA strand?
Mhm. Yep.
How could something make a complementary copy, but if they're both together, if they just do something else?
They split. So it's going to split in half, so a copy can be made.
Then do they go back together? Or?
Mm-hmm. Then they zip back up. And then we're going to have some DNA in here that we don't yet know the function of. So there's a little stretch of DNA that we're like, yeah, I don't really know what that does yet. And then another 1,000 nucleotides makes up a different gene. And each of those genes is going to make a different protein. So when I say that your DNA is nothing but a series of recipes, I actually mean that. Like, I mean that it's just a series of instructions to make a different protein all stacked next to each other. Now you'll notice that part that I underlined, I said, is a stretch of DNA. We don't know what it does yet. When I was your age, scientists called that junk DNA. They said it didn't do anything. Had no function. So we labeled it junk DNA, and then that was it. We just ignored it. It turns out that was pretty short-sighted, because it has functions that we did not understand. So we no longer use the term junk DNA, and we just call it DNA that we aren't sure, we don't know, or have no evidence of its function. So what one's bigger? Gene or DNA? Which one makes up which one? Genes are made out of stretches of DNA. The definition of a gene, by the way, the technical definition, is a series of nucleotides that code for protein. That's what a gene is. Stretch of nucleotide that codes for protein. How do you spell complementary? Oh, there are two correct spellings. Which one means, gosh, I really like your socks? Right? Wow, you look great. That's a compliment. What's the second one mean?
Like, if you add onto something, like they go well together.
They go well together. What about mathematically? Do you know anything about complementary angles?
Yeah, they add it to 90 or something. Right?
They add up to? So they add up to 90? So that means that they add up, or they go well together. If you have a friend who compliments your personality, they're not saying, wow, you're really optimistic. It means that you together as friends work well together, right? I say this because this leads to some confusion. Mm-hmm. We start labeling the carbons by starting at the carbon that's bonded to the nitrogen base. Don't move in the direction of oxygen. You can't label oxygen. So you move in the opposite direction and label every carbon that you encounter in the line. Because of that, we label this side the 5', that's how you read apostrophe in math and science, 5' end of the nucleotide. That's the 5' end of the nucleotide. The opposite looking at the one next to it, I start 1, that's the first carbon. I don't move in the direction of oxygen, I move in the other. So here's 2, 3, 4, and 5. So this side is labeled the 3' side. This seems like the world's tiniest detail, right? You're like, wow, that's an atomic detail. Quite literally, it is an atomic detail because it's about an individual atom. It's about carbon atoms. Seems like a truly tiny detail, but it is absolutely essential you know that to understand DNA replication. You can't understand how we can make a direct, exact copy of DNA if you do not know that there's a 5' carbon and a 3' carbon. Because I'm going to throw that term out there, and you're going to be like, what? Mm-hmm.
What's the difference between them?
It's where they're oriented. So it's like giving them a cardinal direction or saying north, south, or saying top or bottom. But instead of saying top and bottom, we label them 3 and 5. So here, on this one, I say 1, 2, 3, 4, 5, and that's how I end up with the 5' end. Here is the 3' end, because the third carbon is the one that was on that end, that side. What you need to know about DNA, the double helix, they are what we call anti-parallel. Their backbones run in opposite chemical directions, 3' to 5'. So you'll notice here, 5' on the bottom. The strand that's complementary to it, 3' is on the bottom.
So the backbones run in opposite directions?
So unfortunately, sometimes we think about these as parallel lines, right? You guys think of parallel lines running in the same direction, never to cross. That's true on the DNA backbone. To our picture, don't think of the backbones as, guys, focus. Don't think of your backbones as lines. Think about them as vectors. Notice the arrow. One goes down, the other goes up. In DNA, the vector points in the 3' direction. So on the backbone, it points in the 3' direction. So we call that anti-parallel.